In this lesson, you will have one video that I'm going to teach you writing on the board what you need to do. I would like you to copy everything that I'm writing on the board. That means that you need to have a notebook. You will have uploaded one PowerPoint presentation that includes all the information useful for study from. You will have a test by the end of the lesson and a resource for your parents if they would like to examine you in some way. So the main difference between the prokaryotic cell and eukaryotic cell is this compartmentalization of the eukaryotic cell. As we said before, the prokaryotic cells, the prokaryotic organisms are those who first evolve on Earth, and always the first one is the most simple one. The prokaryotic cells are simple cells, and evolution went on going to eukaryotic cell. The compartmentalization during this process of evolution shows some advantages. And what are the advantages of compartmentalization? We are going to find out now. Advantages of compartmentalization. The first one advantage, enzymes and substrate are not spread through the whole cytoplasm. They are located on specific place in specific organelles. Enzymes and substrates are located in specific locations. In the cytoplasm. Which are these locations? Which are these organelles? We are going to talk in this lesson later on. Enzymes, we talked about enzymes. These are the biological catalysts, those who speed up and regulate and coordinate the activities of the cells. These activities are actually chemical reactions, and these chemical reactions are called in biology with the word substrate. So enzymes and substrates are not spread around the whole cytoplasm. They are located on a specific place, which actually is a very big advantage, which brings to the function of the cell. Each one function of the cell is much more regulated and backed up that it's gonna occur in a certain specific way. The second advantage of compartmentalization is that the harmful substances are kept, just like the enzymes and substrates, are kept away by the membranes that surround the organelles. So if there is something harmful within the cell, it stays in specific structure, in specific cell organelle, so they cannot spread all over the cytoplasm. It cannot poison or harm the whole cell. The other advantage of compartmentalization is that the pH of different organelles is kept at particular levels which are relevant to each organelle. pH of the organelles is kept on certain levels, and these levels are characteristic, relevant to each cell organelle. Let's say that, talking about Golgi apparatus as a cell organelle and endoplasmic reticulum as a cell organelle, they both are membrane organelles. Some chemical processes occur in the both organelles, but the chemical processes that occur in the both organelles are very different from each other. As a result of these chemical processes, the pH of each of these organelle is different. So this compartmentalization, this membrane that bound the both cell organelles, makes sure that the pH of those two organelles is going to be relevant to the organelle, relevant to the requirements of their functions. And the last one advantage is, there are many advantages, but the last one main advantage is that organelles with their content can move around within the cell without harming the cell. Organelles can move around the cell. They just float in the cytoplasm of the cell, and that doesn't make the function of the cell to be harmed. Each one cell organelle has specific function, and the organelle and the cell cannot survive without it. Some of the cell structures, some of the cell organelles are temporary, some other organelles are permanent. The permanent organelles are very important for the cell, and we're going to start with the cell as with the nucleus as a cell organelle, because it is very important for the overall function and survival of the cell. So the first one organelle is the nucleus. The first one organelle that we are going to talk about is the nucleus. So let's see what about its structure. What about the structure of the nucleus that we need to understand and to memorize? You need to know that it is surrounded by a membrane. It contains chromosomes consisting of DNA. And it also contains chromatin. So here is one image of drawing of a nucleus. What we need to know about the nucleus: it is surrounded by a membrane. As you can see, there are small holes, small pores on the nuclear membrane, which means that the nucleus by itself exchanges substances with the cytoplasm, with the other parts of the cell. It cannot exist by itself because some chemical reactions occur inside, some vital processes occur inside, and this nucleus should exchange some properties, some substances with the environment. In this case, the environment of the of this nucleus is the environment of this nucleus is the cytoplasm. What we have inside of the nucleus? One small structure which is not a membrane bound, it is not surrounded by a membrane, and it is called nucleolus. Nucleolus means, in Latin, means small nucleus. What is the role of this nucleus is still not very clear. It takes part in the division of the cell later on, when the cell needs to be divided into two daughter cells. What else we have inside is the so-called chromatin. All the cell inside is not dry, it is full, just like the whole cell is full of cytoplasm. The whole nucleus is full of some jelly-like substance which is called chromatin. Here, these small drawings are actually the chromosomes, the DNA molecules. We cannot see chromosomes right now because the chromosomes could be seen as distinct structures just when the cell enters into cell division. If the cell is in rest, the chromosomes could not be seen as structures, but all the time the nucleus contains the DNA molecules. These drawings inside are the DNA molecules. So let's see what are the functions of the nucleus. Functions of the nucleus. The first most important function of the nucleus is that it's storing the genetic material. Storing the genetic material. This genetic material, when the science talk about the genetic material from the point of view of biochemistry, just what we are doing right now, this genetic material is called DNA. But if we talk about from the point of view of genetics, we call this genetic material chromosomes. Chromosomes and DNA molecule is the same thing. Another thing that is very important for the function of the nucleus is the so-called DNA replication. This replication happens when the cell goes into cell division. The genetic material, the DNA molecules, are always a certain number per specific species. For example, the genetic material of a human being consists of 46 chromosomes, 46 DNA molecules. In these molecules, a specific information is written inside. For each one molecule, there is a specific information. Each one molecule is responsible for a specific information, which means that when the cell divides, the two new daughter cells should be absolutely identical. Should be identical, having the same genetic material. That means that just before the cell division, the genetic materials need to replicate exactly, very perfectly. And the other one function of the cell is the so-called transcription and messenger RNA information is stored over there. Of course, we are going to talk about transcription and translation later on, but that it's actually, what does it mean actually, is that when we have two new daughter cells, it's not enough just to receive the identical genetic information. This information should be read correctly, and later on to be realized correctly. Or transcription and translation means that the new daughter cells should synthesize identical proteins. Synthesizing identical proteins means that the new cells have read the genetic information exactly as it appears within the nucleus.